So today we're going to talk about genome-wide association studies. And with genome-wide association studies, really our goal is to take single nucleotide polymorphisms, so single substitutions of alleles in the genome, and associate them across the genome with a phenotype of interest. Genome-wide association studies are particularly useful for complex traits. So traits that have both a genetic and an environmental component, and that genetic component is made up of really many different genes or positions across the genome, and those genes might have very subtle effects on your phenotype. GWAS really gets at the common disease, common variant hypothesis. And so in this figure here, from 2008, I have along the x-axis the allele frequency going from very rare variants to common variants. And then along the y-axis here is the penetrance or the effect size. And with Mendelian diseases, right, we are in this upper left-hand corner where we have very rare variants that are have a very large effect on your phenotype. With GWAS, right, we're all the way on the other side of this graph where we are looking at common alleles, and we're expecting low penetrance or low effect sizes on our phenotype. GWAS have been performed for over 10 years now. It actually began with the HapMap project that was found common genetic variation across humans and in a number of individuals by sequencing. And that variation that was found through the HapMap project was initially put onto commercial genotyping arrays. And these arrays allowed you to get known genetic variants that varied in humans and then assay them in your study and in your individuals of interest. And so the goal is to take this set of genome-wide markers and look for association with an outcome of interest. As Chris said, GWAS have been very successful at identifying SNPs across the genome associated with various traits. Here is a figure from the GWAS catalog. The GWAS catalog, it aggregates the genome-wide significant associations between SNPs and traits. And from earlier this month, the GWAS catalog contained 3,000 publications and 87,000 unique SNP trait associations. So there is a lot that has been found through genome-wide association studies. So we're going to talk about the three main steps in performing a genome-wide association study. We're going to talk about how to design a genome-wide association study, some considerations, quality control that should be undertaken, and then analysis. And I'm going to talk about the first analysis doing a GWAS, as well as I'm going to mention some follow-up and secondary analyses you'll do after you do your primary genome-wide association study. And really, we're only going to scratch the surface here. I'm going to give you the background and give you insights into where you can go further and take this the next step. So the first thing you have to do when you want to do a genome-wide association study is you have to identify your outcome of interest. What are you interested in studying? Are you going to use a disease trait? So cases controls, like type 2 diabetes cases versus controls, schizophrenic cases and controls, or obesity, yes or no? Or you're going to look at a quantitative trait, like BMI, your cholesterol levels, and gene expression. But most importantly, before performing a genome-wide association study, your trait should be heritable. It should have a genetic component to that trait because you're going to be looking for genetic markers associated with that trait. So it's good to make sure you understand whether your trait is heritable or not. The first thing to consider is what kind of a sample size do I need to do my genome-wide association study? This is an actual figure from a paper from 2005, and along the x-axis here is the frequency of the disease susceptibility allele. And then it has, for various effect sizes, here odds ratios, it has the sample size required to achieve 80% power, and this was done for a 10 to negative 6 alpha level. And what you can see, if I go to a frequency of 0.2 for my SNP, and I say I want a odds ratio, I want to be able to detect an odds ratio of 1.2, right? That means I need about 5,000 individuals in my study to be able to detect an odds ratio of 1.2 with a SNP that has a frequency of 0.2. Now, we typically, these days, we don't observe odds ratios this high, right? So we're observing odds ratios much lower than this, say in the 1.02 range or 1.01 range, and therefore we need much larger sample sizes today to detect the small effects that we're detecting within genome-wide association studies. So there's two routes to doing genome-wide association studies. You can use a single study where you've collected all the data, and this is most useful when you have a really unique phenotype that hasn't been collected routinely. On the other hand, you can do multi-study studies where this is considered a consortium model where there are commonly collected phenotypes. And such as BMI, or what I work with, cholesterol, type 2 diabetes typically works in a consortium model where multiple studies are going to contribute to the GWAS results. And this ends up resulting in a meta-analysis of the individual study results to combine the evidence across the different studies. And we'll talk a little more about that in a little bit. The UK Biobank is a recent resource of genetic and phenotypic data on approximately 500,000 individuals from the UK between 40 and 69 years old, and they've collected a range of phenotypic information on these individuals that might be useful. It's a prospective cohort study, and the data is actually open to all bona fide health researchers. You can apply. There's a nominal fee to obtain the data, and you can ask unique questions with this data set about your studies of interest if you're interested. And so the UK Biobank is a great resource and is been used over the past couple of years to do genome-wide association studies in a very large cohort. Now, besides your phenotype, you also have to get genetic data, and you get that through genotyping arrays. There are over 40 different genotyping arrays that have been developed over the past 10 years. They're typically from either Illumina or the Affymetrix platforms. And so you have to decide on the array that you're going to use for your genotyping. Typically, this is dependent on when you've doing your genotyping and the costs of the genotyping and your study design if you're trying to assay a particular set of variants or a particular population of individuals. Now, QC is an essential step in analyzing genetic data. QC steps are undertaken to remove false positive associations. We usually take steps to remove individuals and markers that might have particularly high error rates. Assuming many individuals have been genotyped, removal of a handful of individuals really should have little effect on your overall power. And given the large number of markers genotyped with these arrays, the removal of a small percentage of markers don't markedly decrease your overall power of the study. So here is some of the common QC metrics that are undertaken, where we do both sample QC and SNP QC. For sample QC, we typically look at missingness, deviations from heterozygosity. We'll check to make sure the genetic sex matches the sex that's provided in the phenotypes. We look for duplicates, cryptic relatedness, unexpected relatedness between individuals. We could look for population outliers, people who don't fit with the ancestry we're expecting. And then if we have family data, we can look for Mendelian errors within the data. For the SNP QC, we look for missingness, high missingness, deviations from Hardy-Weinberg equilibrium, differential missingness between cases controls, if we have cases controls, to make sure there's no difference in the genotyping assaying of the cases and controls, as well as we could look at Mendelian errors within the SNP data as well.
Could you just expand on the Mendelian errors? One more sentence about.
Yeah, sure. So so I work a lot with the Framingham Heart Study, where we have families, and so we have two parents and a child, right? And so say my two parents, right? Both my father has a 2-2 genotype and my mother has the 1-1 genotype. I expect my child will be a heterozygote 1-2 genotype, right? If they are something else, if they are a 2-2 child, that doesn't fit with getting one allele from the mother and one allele from the father. So that would indicate that somewhere in the family there's a genotyping error. And you really don't know which individual is the problem. And so in that case, we can look at this on the individual level to see if one particular individual has a lot of errors, or one particular SNP seems to have a lot of those errors. And that could indicate whether that individual is badly genotyped or that SNP is badly genotyped.
How often do you find uniparental guys?
What?
How often do you find UPD, uniparental guys?
Can you repeat the question?
Oh, thank you. How often do you find uniparental diasis? I don't know.
I mean, you have to look at the SNPs that were all in that same chromosome and see if the errors were all.
Yeah, with the Framingham Heart Study, we haven't seen any gross problems with looking at Mendelian errors, yeah.
But it would be the same thing for CNV. If you usually do your QC, and if you find a cluster where you're missing one side, you would be able to, from that itself, know whether or not you have a de novo copy number variation with a deletion on one side, or you need to do another technique to figure that out.
Yeah.
But if we see runs, then you don't throw that individual.
Right, it's a gross problem where you are looking for this.
I have the same question, because you're talking about it as it's a quality control with the sample, and it might not be if it's uniparental disomy. It might be a Mendelian other error, loss of heterozygosity with.
Yeah, but this is across the whole genome,
Gotcha.
right? So we're,
For an individual.
it's not particularly that you're worried about, you're aggregating this to determine a problem across many markers or many individuals. So it's not a particular site. Usually QC is actually performed on samples first and then markers, because you have many more markers contributing to those sample level statistics than the opposite way. So that's typically the order that the QC is done. Sometimes it's gone back and forth between the two as a second round. Now, population structure is really important to understand and take care of in genetic association studies, because if there's subpopulations within your data and you don't know about it, and they differ with respect to both the trait distributions and the marker genotype frequencies, confounding and spurious associations may result. And so this is a typical epidemiological confounding model, where my confounder is population structure, right? I have subpopulations within my data. And even within European samples, it is known that population structure exists, where there subpopulations exist, where that the populations or subpopulations are associated with your genetic variants of interest, and your subpopulations are associated with differential prevalence of your outcome. You can get a spurious association between your genetic variant and your outcome of interest. We've, over the last 10 years, been using these principal components of ancestry, so aggregates of weighted aggregates of SNPs across the genome to get at individuals' predicted ancestry relative to each other. And so this is a principal component plot that just shows how people can use these principal components. Each dot is a person, and you can see that doing this will kind of segregates out the Italians from the others in this plot. There's other ways to control for population stratification. You can use well-matched study designs. It could be you can use these principal components as adjustments in your association test. And a newer approach is using mixed models, using a genetic relationship matrix to adjust for both for cryptic relatedness, known relatedness, as well as a structure within your data. So I'm just putting up a couple of references and resources on best practice for quality control, because I just kind of gave you an overview of different things we look at for quality control, and there's these great papers that really go into different how to quality control your genome-wide association analysis. So when we do a study where we're combining multiple studies and doing a meta-analysis, we typically do this to improve power by increasing our sample size. So allowing us to have multiple studies contributing to the genome-wide association study. The problem with doing a meta-analysis is that different studies typically use different commercial chips with different sets of SNPs on those chips for their genotyping arrays. So when studies use different sets of SNPs, common SNPs can be analyzed together, but this set of common SNPs across platforms is typically relatively small. So here's an example of this. Here is a reference sample that has genotypes on many SNPs. Here's our sample that has genotypes of SNPs from our array that we used. So we can use imputation to fill in the missing SNPs based on a set of reference haplotypes. So we can infer those missing SNPs from our sample and that were not typed and obtain estimates or inferences of those missing SNP genotypes. And this allows us then to use this in our meta-analysis across different platforms that were used for assaying genotypes. Now, there are multiple imputation platforms available, so the haplotypes for imputing SNPs. The original one was the HapMap project, had the reference haplotypes. That expanded to be the 1,000 Genomes Project haplotypes. And then that expanded to the Haplotype Reference Consortium haplotypes. And then the imputation panel, the latest one is the Transomic for Precision Medicine, or TOPMed panel. And these are all built off of whole genome sequencing data. There are multiple tools for doing imputation, and here are the four most popular tools for imputing missing SNPs from reference haplotypes. And I want to say Mock and Impute2 seem to be the two most popular ones. After you use these software to get imputation, yeah?
Very naive question. When you're imputing the SNPs, how do you deal with polymorphic loci? If it's polymorphism in the area, how do you
Right, so
Could you repeat the question?
how do you deal with polymorphism in the area of the SNP?
Yes.
Right, so it's an algorithm, right, that is taking into account these imputation softwares. They're taking a Bayesian approach for determining what the possible genotype is at this SNP. And actually, when you get these imputed values, they range from 0 to 2. So they actually give you a probabilistic kind of information, and it's not necessarily you have 0, 1, or 2 copies of the alternate allele, right? Maybe you have 1.5, because the algorithm couldn't precisely tell you it's a 1 or a 2, but somewhere it's most likely one of those two. Yes? Okay. Also on imputation, right? You get a quality measure of your imputation. For each SNP that's imputed, you get how well it's been imputed. These quality measures are slightly different dependent on which software you're using, but a value close to 1 indicates good quality of the SNP. And typically SNPs are excluded if they have low imputation quality, so less than 0.4 or 0.3, meaning that the algorithm that was used didn't quite impute that SNP well. So going back to my example of having 2 different studies with 2 different arrays that were used, and we want to combine them together, right? So we have study 1 who had an Affymetric array, and study 2 that has an Illumina array. Before imputation, say in this little region of the genome, there is actually only 4 SNPs that are in common between the 2 arrays, right? So that means if I really wanted to analyze just the genotyping data, I would only be able to analyze these 4 SNPs. But after imputation of these 2 studies, right, I fill in all the gaps, and I'm able to analyze many more SNPs across this region. So imputation is filling in the gaps based on a reference panel. Questions? All right, so that's what I had to say about study design and quality control. So now I'm going to start talking about analysis of GWAS. I'm going to start with just what you do for performing primary GWAS, and then I'll briefly go through some follow-up and secondary analyses that can be done. So here I have just a simple regression model where I have some trait value yi for each individual I, and I have their independent variable xi. I'm going to just say xi equals 1 if an individual I has the A allele and 0 elsewhere. Right? And so this is just a simple linear regression model. And with this, what I'm doing is I'm basically testing the mean difference between having the A allele and not having the A allele. So I have the phenotypes for those with the A allele and those without the A allele. And I'm really getting with that beta this difference in means here between these 2 groups. And so that's what we're trying to do. We're going to do this, and we're going to do this for each marker across the genome, and we want to, if we have a quantitative trait, right, for each marker, we're going to compare the trend in the trait to the trend in the genotypes, and we can do that with a linear regression model. If I have a dichotomous trait, for each marker, we're going to compare the allele frequency in the cases to the allele frequency in the controls. And there's multiple methods I can use for that. I can do a chi-square test, I can do a Fisher's exact test, a trend test. If I want to adjust for covariates, I can use a logistic regression model. But the model you use depends on what your outcome is. You can control for covariates, so age and sex, or other covariates that might be influencing your outcome. We typically also control for possible population structure using principal components of ancestry. And if you have related individuals in your data, you can use a genetic relationship matrix to control for that. So when we're performing a test of an association, what we're doing is we're testing the null hypothesis, that there's no association between each SNP and the outcome. And we're testing that through looking at whether our null hypothesis, our beta I value, so for the individual SNP, is equal to 0. And then we're comparing that to our alternative hypothesis, that there is an association between SNP I and our outcome, so that beta I is not equal to 0. From this, you get 3 pieces of information. You get your effect size, so either your odds ratio if you're doing a logistic regression model, or beta for linear regression. And your effect size really tells you how much an effect does the genotype have on the outcome. The standard error gives us how much variability is there in this beta estimate. And our p-value is what's the significance of the association. Well, the last part is, what is an appropriate significance level? Well, with genome-wide association studies, we're testing many SNPs for association with your trait of interest. And the simplest way to handle this is this multiple comparisons or multiple testing is to correct our overall alpha level to allow the family-wise error rate, so our overall study rate, to be close to our 0.05 level. And the simplest way to do this is to use a Bonferroni correction. So divide the overall alpha level by the number of tests and compare the p-value to an adjusted level. And so that's typically what is done. Now, in GWAS have been typically using 5 times 10 to the negative 10th. And that is because it's a correction for about 1 million independent tests. So there's in European populations, there's about 1 million independent common SNPs that have been estimated. And that's what we're using for our threshold when we test common variants. Now, this does not translate to other populations with other LD structure. For example, this threshold will have to be more stringent when testing African Americans because their LD structure is different. And it also does not translate to when you start looking at rare variants, right? Because there's more variants in the rare variant space. And so we're going to actually have to be more stringent with rare variants than we are here. So when you have multiple studies contributing to your GWAS, there's 2 approaches that you can take. You can combine the individual level data from the cohorts into one big squared off matrix for analysis. Oftentimes this is not feasible to transfer the data because of patient confidentiality. It also might not be statistically wise to do this, given that there's different designs of the different studies, different quality control or arrays that were used. Even the genotyping could cause problems with combining the data together. So a typical approach is actually using meta-analysis by combining evidence of association across studies. And so from a meta-analysis perspective is you're doing GWAS within each of the individual studies, and then you're statistically combining the evidence from all those individual GWAS and to get one single GWAS estimate of effect. And so you can combine test statistics, p-values, or effect estimates. The most popular approach is an inverse variance weighted meta-analysis, and it's implemented in most GWAS meta-analysis software. It's often referred to as the fixed effects meta-analysis. And here you get a weighted average of estimated effects from each study, taking the precision of the effect estimates into consideration, so that larger studies are more precise and are given more weight, and smaller studies are less precise and have are given less weights, and these weights are inversely proportional to the standard error squared. So just to put this up, say we have beta k estimates from k different studies, and we have SK standard errors from those k studies, right? So you have these GWAS in each of the individual studies. We can get weights, which is just inversely proportional to the standard error squared. So weights for each study. We can get a pooled effect estimate over all the studies, right, based on this formula. We can then get the standard error for that pooled effect estimate. And then we can calculate a z-statistic, which is just the effect estimate over the standard error. And under the Null distribution, this z-statistic is normally distributed. And so we can calculate p-values based on that. So this is the simplest way to do meta-analysis. And here are some references and best practices on meta-analysis. And the point I want to make here is that it's important to actually perform QC of those individual level study results files before you do meta-analysis. And there's this software called EasyQC that really makes it straightforward to be able to do the quality control before doing the meta-analysis. And so this program generates nice QC descriptives before you do your meta-analysis so you can filter variants that might be problematic. So when you do a GWAS, right, after you do the meta-analysis or after you have your study results, right, you get basically over 2 million association results. You get a flat file that for each line is an association of some SNP with your outcome of interest, right? And so that's a really big file. And so you need to be able to summarize that in a way that's meaningful. And there's 3 different kind of plots we traditionally do to summarize this kind of associations. The first is the QQ plot, where you plot the expected log p-value against the observed log p-value. And this, under the null, under no association, you expect that the dots, each dot is a association results, right? It's a minus log 10 p-value. You expect the dots to follow this 45-degree line. And that indicates that your study test statistics are well behaved. And you can also calculate something called the genomic control lambda value, where you take the median chi-square statistic, the median observed chi-square statistic, and divide it by the expected median chi-square statistic. Now your median chi-square statistic is somewhere down here, and that's where actually the bulk of your points lie, is down here in the distribution. What I have up here is I have something that is deviant from the distribution, and this indicates that there probably is a true association here with this SNP. But you expect genomic control lambda value, you expect it to be close to 1. If you have a genomic control lambda value greater than 1, it indicates an inflation in test statistics, and less than 1 indicates a deflation in test statistics. This plot here, you can see that I have some systematic inflation in my test statistics here, right? It's deviating off the line much earlier than expected, and this could result from unaccounted systematic differences between cases and controls. It could be due to population structure. It could be due to unaccounted for relatedness, technical bias, or poor quality genotypes. So when you have something that looks like this, you need to kind of look into it further to determine what's going on. These are some real QQ plots. Those other ones I had simulated the data. And you can see here I have from a recent LDL GWAS. And you can see right here is my 45-degree line. You can't even see it because we have such significant results. And because we've been analyzing this data and increasing our sample size so much over time. And here we've removed these known SNPs, and we just look at, we can then parse out these, the 45-degree line even better. Here is a Manhattan plot, another way we summarize genome-wide association studies. Here, once again, each point indicates a SNP association, right? And what you have is along the x-axis now, you have the position of that SNP in the genome. And then along the y-axis, you have the minus log 10 p-value. And so it gives you a view across the genome of your associations. And you expect these peaks because of LD. So because SNPs are in LD, you expect these kind of these peaks to occur for the associated SNPs. The third plot is really narrowing down. It's taking this Manhattan plot and really focusing down in or zooming into a particular region to be able to look at that region more closely and to see what's going on in that particular region. And these are generated, this locus zoom plot can be generated from this website, and you can use it with published data, or you can use your own data and put it into this website to generate these plots. One point I want to make is that association is not causation. That variance, the variant that we are analyzing can have a functional effect on the trait. It could cause amino acid change. It could cause a stop. It could be regulatory and change the amount of protein produced. Or that variance could be in linkage LD with a functional variant. And through association, we can't tell the difference between these two scenarios. Here are some tools, common tools that are used for performing genome-wide association studies. But what I really want to do is spend the last couple minutes talking about what else can be done once you have your associations between your outcome and SNP. And so there's common statistical analyses that are done once you have your GWAS association done. You can estimate the variance explained by a set of SNPs. You can try doing fine mapping, where you try and determine what the independent SNPs are or what the potentially causal SNPs in the locus are. You could look for pleiotropy. Does a SNP relate to multiple traits? Risk prediction. Does a score of genetic markers predict risk for disease? There's Mendelian randomization, where we can leverage genetic markers to get at causality of biomarkers on a particular outcome, as well as pathway analyses. Are the associated loci linked biologically? Here's an example from Height, where they did a Height GWAS in about 130,000 individuals, and then again later on in 250,000 individuals. And you can see that many more SNPs and loci have been identified with increasing the sample sizes, and that they explain a larger proportion of variance in their trait compared to what they found previously. So that when they're increasing their sample sizes, they're finding more loci, they're increasing the variance explained of their traits, as well as the overall explained heritability. Fine mapping is looking at what are the independent SNPs in the locus. This is a locus zoom plot around a particular region that was of interest in a Height GWAS. And what they did was, this plot highlights what is significant after doing a conditional analysis. So they conditioned on the SNPs and then saw how that changed the significance and effect sizes of the SNPs in that region to be able to determine which SNPs were independent within the locus. Identifying causal variants statistically that underline associations has been particularly difficult. There's two kind of key steps in determining causality of SNPs. The first is getting well-calibrated probabilities of causality through Bayesian methods to find the candidate SNPs, and then trying to connect these variants to likely genes whose permutation may lead to altered disease risk using functional annotations. So there's some ways we can get at causality within a locus, but statistically, but really you need functional experiments to get at that. Pleiotropy is getting at, do SNPs relate to multiple traits? So here's an example from lipids, where we had 157 lipid loci, and we then took those loci and looked at the GWAS from coronary artery disease, BMI, blood pressure, waist to hip ratio, systolic blood pressure, type 2 diabetes, and fasting glucose, and looked at a enrichment in association results in these other GWAS for our lipid-associated variants. And we saw some enrichment of different excess for these different traits. MTAG is a method that was published about this time last year for doing a joint analysis of summary statistics from GWAS summary statistics and being able to model that appropriately. One can also take the beta estimates from a GWAS and create genetic risk scores and use these genetic risk scores to determine whether score genetic markers predict risk of disease. So for each individual J, you can calculate this genetic risk score, which is just really a sum of SNPs times their weights, their betas, from the GWAS. And then in this study looked at a series of different genetic risk scores for using different trait weights and different number of SNPs against these 7 different outcomes to see how these genetic risk scores predict these outcomes. And they found really that the genetic risk score based on the GWAS did best at predicting the trait from which the GWAS was done from. All right, Mendelian randomization. Mendelian randomization can be used to leverage genetic variance to get at causality of biomarkers, and this is akin to doing a randomized control trial because at meiosis, your alleles are randomly given to a gamete, and so there's this randomization step when you, during meiosis. And so we can use this to get at causality of biomarkers. For Mendelian randomization, the assumptions are that the genetic marker is strongly associated with your exposure that you want to test for causality of. It's independent of the outcome given the exposure, and then it's independent of factors that confound the exposure-outcome relationship. And you can do this instrumental variable or Mendelian randomization approach using summary statistics from GWAS. And this is called the Toby Johnson method for risk prediction. We've done this in lipids, where for LDL, we found that genetically elevated LDL increases your risk for heart attack, which was expected given clinical trials. But for HDL, those with genetically elevated HDL were not protected from MI. There's multiple pathway analyses that have been done. I believe all of these have been developed by Brody's. So different pathway programs and methods for seeing whether your results are aggregating in a particular pathway. We did this for lipids with MAGENTA and was used to evaluate the overrepresentation of biological pathways among our lipid loci. And among the 57 loci, we found 71 enriched pathways, and these made biologically sense for our lipid loci. So in summary, GWAS have been successful at locating regions of the genome with associations with complex traits. Many of these loci that are found with GWAS are non-coding and don't have known gene function. There are many more genetic variants to be found. We're finding that as we increase our sample sizes for common variants, we're getting more associated variants just with smaller effects, but those have been helpful in these downstream analyses, stream analyses that we've been doing. And you know, the GWAS to date have been predominantly done in samples of European origin, and so there's still left a lot to be found in other population groups. And then finally, there's many types of analyses that can be done based on summary statistics, either after you've performed your GWAS or on publicly available GWAS summary statistics. So that's all I have for today. Thank you. I can take any further questions.
That was a great lecture, thank you. As you start to look at moving forward, everybody that has populations has probably done SNP genotyping, maybe gone to whole exomic sequencing, and is on the verge of going to whole genome sequencing, where many of the association problems just get magnified because the number of associations are going to go sky high, but the end of the population comes shrinking down because people aren't ready to do this yet for the cost on a large population. So how many of these approaches that you had to move from association to causation are going to find a nice home now in whole genome sequencing to try to find causation to the ones they have, and how useful are they going to be to sort out the now thousands of variables you have, but with much smaller numbers? Your power of GWAS is sort of castrated here in one sense, but the thinking behind each one of these is very similar.
So,
Could you repeat all that? Castration?
I think the question is
What's the
somewhere,
applicability for
yeah,
whole genome?
Yeah, so the applicability of the question is using these secondary data techniques for whole genome sequencing. And so I think it's great that we're going to have more variance and we're going to be able to capture everything.
Says you.
Yeah, but I think those secondary analyses have only become more powerful and gained more insights because of the large sample sizes that we have in GWAS, and that we get pretty precise now estimates of effect. So everything you do, secondary analyses, right, relying on that GWAS, you're relying on that GWAS effect estimate to be right? So you have to have precise estimates in that GWAS effect for those GWAS effect estimates to have the most power when you're doing those secondary analyses. So I think it's going to be hard when we have these small sample sizes in whole genome sequencing, but as we increase these secondary analysis techniques will become more powerful. Yeah. Right.
Great.
Thanks.
Thanks very much.